Hello everyone, my name is Camryn Bonn, and I will be examining the mechanism of FarR in promoting increased resistance of Staphylococcus aureus to antimicrobial-free fatty acids on human skin. Staphylococcus aureus is a gram-positive bacterium that forms clusters of cocci. Approximately 30% of healthy adults are asymptomatically colonized, preferentially in the nares but also on the skin. Typically, the colonizing strain most commonly causes subsequent infection. Consequently, S. aureus is opportunistic and utilizes immunocompromise to cause invasive infection, such as soft tissue abscesses, lower respiratory infection, pneumonia, endocarditis, bacteremia, septicemia, osteomyelitis, and cystic fibrosis complications. Burden on human health was also increased with the introduction of antibiotic resistance in 1961 through the acquisition of the mobile element SCC-MEC. Methicillin-resistant S. aureus, or MRSA, is prevalent in two forms. The most common community-acquired, or CA-MRSA strain is USA 300, possessing the arginine catabolic mobile element ACME, and SPG for polyamine resistance. On the other hand, the most common hospital-associated, or HA-MRSA, is USA 100, possessing vancomycin resistance and lacking ACME. USA 300 represents our laboratory strain, while USA 100 represents our clinical strain. As previously mentioned, skin/soft tissue infections are important and represent 90% of all CA-MRSA, indicating a need to examine S. aureus colonization of the skin. The skin represents the initial defense against pathogens, utilizing keratinocytes with tight junctions, antimicrobial peptides, immune cells, and a competitive microbiome to protect against infection. However, it is the acidic pH of 4.5 to 5.9 that is of particular interest. As observed in the diagram, many mechanisms contribute to pH, such as the conversion of filigran to uronic acid, the NHE1 sodium proton antiporter, proteases, and enzymes. Today, I'm going to be focusing on the conversion of phospholipids and ceramides from sebum into unsaturated free fatty acids, or UFFA, by commensal secretory lipase A2, or ceramididase. These UFFA are toxic to bacteria through membrane insertion that causes subsequent instability and increased fluidity. Protein leakage, electron transport chain interference, and dysregulated nutrient uptake can cause cell death. However, these UFFA aren't always toxic. S. aureus has used these lipids to its benefit in its first line of defense against toxic UFFA. UFFA, such as sapienic acid in sebum and linoleic acid on the mucous membrane of the nares, cross the bacterial cell wall/membrane and enter the fatty acid kinase, or FAK, pathway. Instead of incorporating into the membrane to cause increased fluidity and detrimental cell leakage, UFFA are bound by FAK-B2, then phosphorylated by FAK-A before they enter either the FAS2 or PLS pathways for conversion into incorporation as bacterial phospholipids. However, what happens when the maximum capacity of FAK-A to incorporate these UFFA is reached? This question led to the discovery of a novel UFFA detoxification mechanism in S. aureus. Previously in our lab, a novel TETR family regulator called FarR and a resistance nodulation division R&D superfamily efflux pump called FarE were discovered. USA 300 was exposed to high levels of linoleic acid, which selected for a variant with increased resistance. This variant, called FarR7, possesses an H121Y substitution in the ligand binding domain of FarR, which will be discussed further later. The most well-known example of a TETR family regulator and R&D pump interaction is the ACR-ACR-AB interaction in E. coli. The TETR regulator ACR represses the ACR-AB R&D pump in complex with Tol-C, allowing for the efflux of bile salts, a compound that is similarly hydrophobic to UFFA. Similar to FarR7, mutations arise in ACR during selective pressure caused by antibiotic treatment. The result is de-repression of the efflux pump and resistance to structurally dissimilar compounds such as tetracycline. Based on existing models, we suspect that a FAK-A metabolic product interacts with the ligand binding domain of the TETR family regulator FarR. FarR then binds as a dimer to the divergently transcribed intergenic segment with FarE at 3 operator sites possessing the TAG-WTTA consensus sequence. We suspect the binding of the helix-turn-helix motif of the N-terminal ligand binding domain of FarR at the palindrome PAL2 operator site induces FarE expression and subsequent fatty acid efflux, unlike typical TETR family regulators, which are found to repress their effectors. However, the specific metabolite that causes FarR activation remains unknown. Although resistance nodulation division fatty acid efflux pumps have been widely characterized in gram-negative species, they remain uncharacterized in gram-positive species. Additionally, the ligands, pathways, and mutations responsible for FarR function remain unknown. Thus, we have two hypotheses. First, we hypothesize that once the maximum UFFA incorporation capacity is reached, a metabolite from the fatty acid kinase pathway forms a complex with FarR for activation. Second, we hypothesize that S. aureus, like the ACR system in E. coli, is subject to selective pressures in vivo that result in FarR mutation and subsequent derepression of FarE. Hence, my first aim is to assess the role of a small molecule ligand in modulating the affinity of FarR for the FarE promoter. I will accomplish this by determining the identity of the molecule and/or molecules that are responsible for activating FarR. My second aim is to determine the significance of in vivo amino acid substitutions in FarR in causing increased resistance. I will accomplish this by assessing FarR variants resistant to both UFFA and other antimicrobial compounds. Beginning with aim 1, a previous electrophoretic mobility shift assay from our lab was performed and demonstrated FarR was unable to bind a minimal 24-nucleotide PAL2 probe, as demonstrated by the absence of complex formation for the OP4.1 segment. The first two lanes in each condition represent FarR binding to its autorepressive sites, while the third represents the activating site at PAL2 that induces FarE. This suggests that FarR nonspecifically binds a longer probe, while we suspect a small molecule ligand is required to be present in the EMSA as a cofactor to specifically bind the minimal probe. Two possibilities exist based on these data. 1. An acyl phosphate, a metabolic product from the FAC pathway, binds directly to FarR. Or 2. The acyl phosphate binds as a complex with additional proteins to FarR. This is similar to another activating TETR family regulator called DHAS from Lactococcus lactis that requires the DHAQ protein ligand to activate the dihydroxyacetone operon. We then performed a homology search in the NCBI database, and it returned a 26% identity to the structure of the dihydroxyacetone kinase regulator DHSK from Listeria monocytogenes. The PDB file of DHSK was then used as a template to predict the structure of FarR using Modeler 9.25. Since DHSK requires a small molecule along with a protein for activation, the structural similarities support the hypotheses that a cofactor is involved. You can see the alignment through PIMOL on the left here, with FarR in blue and DHSK in green. This means that FarR could potentially bind to a complex involving any of FAC-A, FAC-B2, ATP, acyl phosphates, and linoleic acid. More work still needs to be completed with the modeling. The comparison of FarR to DHSK needs to be repeated. As well, we would like to model more similar sequences of FarR from different species before comparing the new models to that of S. aureus FarR. My next steps are to repeat the EMSA with a minimal 24-nucleotide PAL2 probe in the presence of FAC-A, FAC-B2, ATP, and linoleic acid. If a shift/complex is formed at Site 3 or the PAL2 operator, then it can be confirmed that a metabolic product or complex from the FAC pathway is involved in the activation of FarR. From here, we will perform size exclusion chromatography, or density gradient centrifugation, followed by mass spectrometry to identify which specific proteins are forming an activation complex with FarR. Now on to aim 2. As mentioned earlier, in vitro selection of USA300 with linoleic acid resulted in discovery of FAR7, a variant of FarR with an H121Y substitution that demonstrates increased UFFA resistance and constitutive FarR expression. Bioinformatic analyses have revealed several prominent FarR amino acid substitutions in a USA100 background. 11 isolates have been collected that possess a C116Y substitution in the C-terminal ligand binding domain of FarR. These isolates are SPA type 2 from the Broad Institute at Harvard and are representative of a wide range of locations such as sputum, bloodstream, and nares. Since the position of the mutation and substitution for a tyrosine residue are similar between the H121Y and C116Y mutants, we suspect that they will share a similar phenotype of increased UFFA resistance. An initial minimum inhibitory concentration assay of the FarR C116Y isolates in the presence of linoleic acid demonstrate increased growth shown by OD600 and thus UFFA resistance compared to USA300 for all isolates except M0049. Sequencing revealed that M0049 does not possess the C116Y substitution. Thus, it possesses wild-type FarR and should display UFFA resistance comparable to USA300. M0182 was also the only isolate to show increased growth and UFFA resistance comparable to that of FarR7. 4 isolates with an E160G substitution in the ligand binding domain of FarR come from cystic fibrosis or CF patients co-infected with Pseudomonas aeruginosa. CF patients co-infected with MRSA and Pseudomonas demonstrate poorer lung function and worse outcomes. However, Pseudomonas produces a hydrophobic small molecule 2N-heptyl-4-hydroxyquinolone N oxide, or HQNO, that incorporates into the MRSA membrane to increase fluidity and allow antibiotic penetration into the biofilm. We suspect that since the molecular structure and mechanism of toxicity of HQNO are similar to UFFA, the FarR E160G isolates evolved increased resistance by utilizing FarE to efflux HQNO. Additionally, 3 isolates with a P165L substitution in the ligand binding domain of FarR have been collected from Mississippi and include MRSA from a CF patient in Georgia, a CF patient in Cincinnati, MRSA from Denmark/Egypt, and cheese-associated MRSA from Austria. These isolates are of particular interest because the T688 spot type has been implicated in potential chlorhexidine resistance in bovine mastitis in Egypt and Kuwait. Chlorhexidine is a disinfectant used in both healthcare and animal agriculture. Since both mutations are located in close proximity to each other, we expect they will share similar resistance phenotypes. An initial MIC of the FarR E160G and P165L isolates in the presence of linoleic acid demonstrated variable growth and UFFA resistance. Both USA300 and USA100 were used as negative controls. Only HP16, HP67, and HP99 from the E160G isolates, along with DAR948 from the P165L isolates, showed increased growth and UFFA resistance compared to USA300 and USA100. Sequencing will be performed to confirm the presence of the desired mutations. DAR 1874, indicated by the asterisk, was also included as it possesses an H43Y substitution in the DNA binding domain of FarR, because we were curious to see if altering the DNA binding affinity of FarR altered UFFA resistance, and in this case, DAR 1874 showed increased growth and UFFA resistance. Finally, 4 strains with a GAG duplication at glutamic acid 93 in FarR are from the nares of patients from the Broad Institute at Harvard, but have also been found in vancomycin intermediate S. aureus, VISA from Japan, MRSA from human skin in Japan, and food-related MRSA from China. M1510 and M1528 possess spot type 2, while M1516 possesses a new spot type with a Rhydom repeat R83, and M1523 possesses a T1340 spot type. An initial MIC of the FarR GAG duplication isolates in the presence of linoleic acid demonstrated increased growth and UFFA resistance compared to USA300 for all isolates, although growth and resistance was not as great as FarR7 or the FarR C116Y isolate M1545, as indicated by the asterisk. Sequencing will again be performed to confirm the presence of the GAG duplication at position 93 in FarR. In summary, the FarR C116Y and GAG duplication isolates showed increased UFFA resistance, while the results for the E160G and P165L isolates were variable. After confirming the sequence of the FarR variants, my next steps include cloning one of each variant into PLI50 for expression in E. coli. From here, I will perform a Western blot using mouse anti-FarR antibodies to determine if, like FarR7, constitutive expression or de-repression of FarR is occurring. The PLI FarR variants will then be cloned into our lab strain of USA300, and an MIC will be performed in the presence of linoleic acid to determine if each amino acid substitution alone is sufficient to cause increased growth and UFFA resistance. Both the original isolates and USA300 PLI FarR constructs will then be subject to MICs with HQNO and chlorhexidine to determine if FarE and FarR provide resistance to alternative antimicrobial compounds. MRSA still represents a significant burden on global health, representing 74% of global S. aureus cases and possessing a bacteremia mortality rate of 30 to 50%. Understanding the mechanism by which FarR is activated and induces efflux of toxic UFFA will provide insight into potential targets to prevent colonization. With persistent antibiotic resistance, it may be beneficial to shift treatment from curative to preventative. Additionally, mutations have arisen in a clinical setting that allow S. aureus to evade innate immune defenses such as UFFA, demonstrating an important example of in vivo selection and adaptation to both hosts and, potentially, to competing microbes such as Pseudomonas. Potential resistance to compounds such as HQNO will provide insight into alleviating the burden of disease in patients with cystic fibrosis, while examining chlorhexidine resistance will benefit our understanding of sanitization procedures. From here, I'd like to thank my supervisor, Dr. Martin McGavin, my supervisory committee members, Dr. David Heinrichs and Dr. John McCormick, past and present lab members, Robert, Catherine, Emily, Gary, Suraj, and Olivia, my clinical isolate suppliers, Dr. Ashley Robinson from Mississippi, Dr. Anthony Fisher from Iowa, and the Broad Institute at Harvard, and finally, Peter for helping me to learn Python to do some protein modeling.